Okay, thanks for coming. Let's get started. I am Mia Sung. It's my pleasure to introduce today's speaker, but before I forget, today's CME code is 50116. And the people who are watching the talk on videocast can submit your questions through the button, and the button says Send Live Feedback. So you can ask the speaker questions that way. Okay, so let me try to give a good introduction because it's really not an easy job to condense Wendell Lim's accomplishment into a short introduction, but I'll do my best. So Dr. Wendell Lim is Bayer's Distinguished Professor at UC San Francisco, Department of Cellular and Molecular Pharmacology. He got his bachelor's degree from Harvard in chemistry and PhD degree at MIT in biochemistry and biophysics. He did his postdoc at Yale, also in biophysics and biochemistry. And then he joined UC San Francisco in 1996. And since then, he has taken numerous leadership positions, including the director of UC San Francisco Center for Systems and Synthetic Biology, which was one of the NIH centers in systems biology until 2019. Since 2020, he has been the director of Cell Design Institute and also the director of Center for Synthetic Immunology, both at UC San Francisco. And beyond UC San Francisco, he has been really serving the entire community of people who are doing quantitative systems biology and synthetic biology in this annual conference series we call Winter Q Bio. He's been a long-time co-organizer in that conference series. Wendell is really a pioneer in synthetic biology approaches, and he has opened multiple different research avenues. So I'll just try to cover a little bit here. So his approaches to engineer immune cells is really cool in the sense that you basically tell lymphocytes to, you tell instructions like if you see A and B, do C and D, such as killing the target cells or activate some genes. So these are really high precision therapeutic approaches that he has pioneered. And in 2016, he published not one, but 3 Cell papers that really demonstrate that this works. With the system that he termed synthetic Notch receptors, or also called SynNotch systems. And in 2022, he published 2 papers in Science about how these CAR T-cell phenotypes can be manipulated by mixing and matching of different signaling motifs or domains. Really beautiful work. And he has so far published about more than 150 peer-reviewed papers, and he has over 60 patents. And I was curious to see what is the top-cited paper out of all this. So I found that it's actually this Cell paper published in 2013, together with actually another Wall speaker next month, Jennifer Doudna, showing that CRISPR can be made to be reversible. So he developed this CRISPR-I method in that paper. So for all of his accomplishments, he has received numerous honors and awards, many distinguished lectures. He has been a board of directors at Burroughs Wellcome Fund. He has been on editorial board of Cell and Science. He's been a Howard Hughes Medical Institute investigator. And even back in 2012, he was named one of the 50 people who will change the world by UK magazine Wired. In today's lecture, we will see how he continues to really push the boundaries, both technologically and conceptually, at the forefront of synthetic biology. Thank you so much for visiting NIH, Wendell. I'd like to welcome you to the program.
Okay, well, can everyone hear me okay? So thank you very much, Mia, for that kind introduction and for inviting me, and hello to everyone both here in person and online. So what I'd like to talk about is our efforts over the last several years to try to ask whether we can actually create new biology. What does it take to actually learn how to program cells and their function, and how can we apply this to various important therapeutic problems? So here are my disclosures. And then what I'd like to do is just start by talking about this general field of synthetic biology and how I think about it. And it's really this idea of learning by building. I think that over the last many decades, with the advent of molecular biology and understanding the gene, we've come to realize that biology and the high-level functions that we see in various organisms are the result of a hierarchy of levels where genetic programs lead to encode molecular networks. These lead to cellular networks, and it's really the ensemble of these behaviors at many scales that leads to the kinds of functions that we all are so enamored of that we see in living systems. And so right now, I think in general, the way that we look at these sorts of systems, where we are interested in a particular function at a high level, and then we tend to try to deconstruct it and map out what are the genes that are involved in this and how are they how do they interact with one another. And this is really focused on taking those biological behaviors that exist and trying to understand them. In a way, what we're interested in synthetic biology is that we're interested in that problem, but really in a bigger context. Maybe another way of phrasing is what are possible? What are things that you could build with biological systems? And that is important from several different perspectives. One is that really, if you look at a broader set of systems and understand what can achieve a certain function, what are the degeneracies, what are the common patterns, the archetypes that you see, I think you can understand, maybe get a better sense of the rules and fundamental principles that govern biological behavior. And then in addition to that, the second thing is that, of course, if you explore this space of using biological components to create new systems, that probably there's some pretty interesting new behaviors out there, some of which could be therapeutically useful. And so we're interested in both of these questions and trying to explore them really in conjunction. And so one way of thinking about that is that, the traditional way perhaps of thinking about problems in immunology or development these are both cases where systems of cells do amazingly complex and precise actions. Perhaps trying to rephrase this in a more generative way in terms of how you create function from the bottom up, we could ask, if we really understand the design logic of these biological cellular systems, how might we be able to, for example, in immunology, engineer immune cells that can precisely recognize and kill solid tumors that normally evade the natural immune system? Or in the case of synthetic development, how might we learn how to program cells so that they self-organize into custom tissues for regeneration or repair? And so by taking on these sorts of problems, we're, again, trying to both do things that we think could be really useful and that only cells could do, but also that can help us give us a bigger context for understanding kind of the principles and rules and logic of biological regulation. The other thing is, right now, we're very focused on the notion of trying to change cellular conversations, how cells interact with one another as networks. And, of course, biology occurs at many different levels. And kind of one of the points I want to make is that, we have made amazing advances at the atomic and molecular scale of understanding kind of the interactions between different modules. Like, for example, the field of chemistry used to be about studying individual molecules. But really now we have this great fundamental understanding of how orbitals and bonds work. And really, chemistry is much more based on principles and not about what molecules exist or don't exist, but what can exist. And just like that, we also understand molecules at a better level. But I think a lot of biology that we look at the higher level really boils down to things that are molecules and other things that are engaging at the cellular level. And so we're very interested in trying to really focus on that. And what we'd ideally like to be able to do is to develop the field of cell engineering and cell design to be as facile as what we can do with today's synthetic chemistry or even protein engineering. Okay, So, and the other reason to focus on cells is that they really do have these amazing capabilities that I think molecules by themselves can't have, whether it's small molecules or biologics. Molecules can, have very precise specificity, but even if you had the most specific molecular drug, because of the fact that these are molecules, they will go everywhere, and because of the fact that the same genes are expressed in different cells, you're going to have systemic cross reactions and toxicities. Cells, however, and this happens, of course, naturally in the body, cells have the ability to sense and integrate environmental information to control where they go and when they execute certain functions. Cells can migrate, they can adhere or be retained in certain places, they can interact very specifically with certain partners, and they can locally induce activities, whether it be proliferation, production of different payloads, or specific communication with other cells. And so I would argue that manipulating cellular agents may be a far more powerful way to interface with diseases, particularly complex diseases that involve sort of cellular ecosystems like cancer, autoimmunity, or fibrosis. Okay, so what I'd like to talk about today, very quickly, is three things. One, I just want to give you an introduction to the type of synthetic biology toolkit that we're developing to rewire cells. Really, what are the fundamental units? If you were going to engineer cells, what are the kind of modules that you need that would give you the most capabilities? And I'll tell you about some of the things that we've been working on and how to use them to rewire how cells interact with themselves and their environment. And then I want to talk about how we can then apply this toolkit to two different problems. Mostly I'll talk about developing next-generation immune cell therapies that really can address solid tumors, but also other tissue diseases beyond cancer, including increased recognition, this development of a tissue GPS system that allows us to tell cells to go to and only act in a specific anatomical tissue, and then also programming cells to treat diseases beyond cancer like autoimmunity or neuroinflammation. And then I'll end with a shorter part to talk about our effort to try to apply this very same tools to a different problem space, that of development, trying to understand how we can wire cells to self-organize and self-pattern into various architectures, and how this might be used to help guide stem cells to develop into specific tissues or shapes. Okay, so let me first talk about this toolkit for rewiring cellular networks. Let's say that we have a problem of a CAR T cell, and we want to be able to engineer it now to go treat a solid tumor. We really need it to be able to talk to the tumor, recognize it, have a conversation with that, talk to the stroma, as well as other immune cells in the endogenous immune system, but also amongst themselves in autocrine sort of ways. And so if we want to do that, what are the words that we need to write new cellular programs to change the relationships and the conversations that these cells have? And ideally, we'd like to be able to, just like with this dry erase marker, pencil in new things and ask, how does that change the behavior of the system? So we want to be able to do that, and we really want to have genetically encoded commands that can program new cellular input-output relationships. And I don't think this is still a difficult but a finite question. That is, we'd like a cellular node to be able to engineer it to detect a new input like X, Y, and Z, something outside in its environment, and then have that turn on a number of kind of canonical responses, whether it's producing a secreted signal or a receptor that allows it to sense something or to change its shape or to divide and grow. They're kind of classical responses that we'd like to be able to have as outputs. Okay, so what are some of these genetic commands? Now, of course, many of you are probably familiar with chimeric antigen receptors, or CARs, which are a really amazing and therapeutically invaluable way to redirect a T-cell response. Basically, it takes the intracellular parts of the TCR combined with co-stimulatory receptors, but then puts on the outside a new input detection system, an antibody for a particular antigen. And that allows us to now redirect the T-cell response to a cell that has this new antigen. So it shows how you can reprogram that input to link it to an endogenous complex output. Now, here's some other things that we have developed or are working on. And one of them, and I'll go in a little bit more detail, is the SynNotch, or synthetic notch system. And just what I quickly want to say is that that's a way we can change the outside with an antibody, a single-chain antibody, to recognize a novel extracellular input. But what it does is that, in the end, it releases a transcriptional domain that goes into the nucleus and turns on the transcription of whatever genetically encoded payload we want that we put behind a responsive promoter. So it's a new way to put in a new input, but then have almost anything as an output that we can encode genetically. So it's very, very flexible as an information transmission modality. And then some other things are what I'll call synthetic cell adhesion molecules, or syncams, that we can change kind of what a cell will recognize, but we can change how strong that interaction and what kind of interface it forms by changing the intracellular signaling domains and how it engages the cytoskeleton. We're also working on things like things that can control trafficking of cells through engineered chemokine systems, but also more recently epigenetic fate switches that can control memory, give you long-term memory as well as fate bifurcation, although I won't talk about those today. Let me just quickly go over the SynNotch receptors. So this is an animation. These were developed by Leo Morsoot and Cole Roybal when they were postdocs in the lab. The natural Notch receptor is a juxtacrine receptor where it detects the signal delta on a neighboring cell. This mechanical force leads to cleavage, and the intracellular part gets cut off, goes into the nucleus, and regulates transcription of various genes. What we showed is that we could actually put an extracellular single-chain antibody to recognize arbitrary target molecule X in the outside world, and then put in an engineered transcription factor on the inside. Now this can go into the nucleus upon activation and turn on whatever genes Y we put behind that responsive promoter. So this allows us to change what we detect in the environment, and now can control also what the cellular response is. Moreover, we have shown that we can change the outsides. We can have different kinds of recognition heads. And then by the same token, we can also change the intracellular part and put on different transcriptional domains that recognize different promoters. And so we can have a whole set of these things that are orthogonal to one another, as well as to the native Notch receptor. And these can all function in the same cell. So that's an incredibly flexible way to change relationships between cells and induce new responses. Very quickly about synthetic adhesion molecules. Adhesion is one of those things that I didn't really appreciate till more recently. Is just sort of ubiquitous. All cells have this. They have to be able to bind to each other to form tissues, to the extracellular matrix. They have to be able to bind to things to migrate or transmigrate through the endothelium. And, of course, they're important for recognition events like the T-cell synapse or even neuronal synapses. And, there are a lot of complex adhesion molecules. What Adam Stevens, a postdoc, showed recently was that we could actually engineer synthetic ones where we replace these complex extracellular domains with, say, antibody-antigen interactions or nanobody interactions like GFP. And then but depending on what kind of intracellular domain we put on the inside and how it engages the intracellular, cytoskeleton, we can get different strength and as well as morphological dependency. So what's shown here is an example just of if we take a GFP interaction, but now have it tied to the intracellular domain of E-cadherin, we get something that actually replicates the kind of broad and tight interface that we see with cadherin. And so this allows us to direct cell-cell interactions, to tune the adhesion strengths and synapse properties, as well as to program multi-cell sorting and assembly. And I'll just show you some movies. These are some things where we've engineered these blue cells with different adhesions, and they, in this case, either can form a node next to the red cells or form this shell around the cluster. So we can start programming those sorts of behaviors. And we've used that in a lot of different ways. OK, now let me talk about, go to the main part of the talk about how we can apply these tools to engineering next-gen immune cell therapies. And of course, everyone knows about CAR-T therapies, one of the first cell therapies that was FDA approved, and in large part due to pioneering work by people like Steve Rosenberg and Jim Korkerdorfer. Here, we know that we can make these artificial receptors that recognize antigens on cancers and kill them, and this has been a remarkable advance for hematopoietic cancers. But we haven't had as much success in targeting solid tumors with CAR-Ts, and that's because it's really not that simple as just recognizing the cancer cells. Recognizing CD19 kills all B cells, and that's actually tolerable, and that's why the therapy can work. We have to have a cell that can recognize the antigens that are on the cancer, but equally important, many antigens are also, especially for solid tumors, are also expressed on some normal tissues. So we have to actually find ways to avoid toxic cross-reaction with these normal tissues. In addition, many of the antigens that are the most tumor-specific are also very heterogeneous. So you can see escape by the endogenous heterogeneity or the loss of the antigens. Certain cells can escape. And so if you get too specific, you also have this problem of more cells might escape. And then also, solid tumors tend to be very suppressive, so you have to overcome the suppressive tumor microenvironment. So the issue is that there are multiple challenges that have to be solved in concert to really get an effective solid tumor therapy. And part of our goal is to try to deploy some of the different modular tools that we have to improve each of these different issues and try to do it in concert. So let me just tell you about one of the solutions that we've been working on for quite some time, which is first focused on increasing recognition specificity of, say, solid tumors, but really, I'll show you, has other implications on some of these other problems. Can we program, for example, a cell, a T cell, to recognize a multi-antigen signature using a two-receptor circuit, where we combine, in this case, SynNotch and a CAR into a circuit? So the standard CAR, of course, might recognize a single tumor-associated antigen in a cancer, but if that's expressed at some level in normal tissues, you could get this cross-reaction that would be toxic. Now, this is what I was talking about. If we could make a cell that has a SynNotch receptor, shown in green here, that would recognize antigen A, that would be in the tumor, and it would be linked to the expression of a CAR gene. So that CAR is not going to be constitutively expressed, but only inducibly expressed. And the idea is, if we make this two-receptor circuit, basally it's off, so it would spare a normal tissue that only had antigen B. But if it is in a tissue that has antigen A, either on the same cancer cell or in a cell that's part of the cancer microenvironment, it would now turn on the SynNotch. This would turn on the CAR, and then the CAR would be able to, would be able to induce expression of the CAR and kill the cells that had antigen B, but only in the case where the A and B were together locally in space. Okay? What's shown here is an animation of what we hoped would happen, and that is that we take these sort of CAR SynNotch T cells, put them into a mouse that had two tumors, one that just had the killing antigen only, and then one that had the killing and the priming antigen. The idea would be that when the cells went in the tumor that only had the killing antigen but no priming antigen, they wouldn't get activated and be neutral. But if they went into the one with the priming antigen in green, they'd get turned on and then be able to kill that only in that case, in that side. And in fact, what we showed that is that we take sort of model tumors, put in one that has the killing, one that has killing and priming. We see this amazing difference in the same animal where the SynNotch CAR T cells only kill that one side. This just shows these are the control T cells aren't killed. If you put in the normal CAR T cells, it'll kill both the tumors, but the SynNotch T cells can really discriminate between the single and the dual antigen. Okay, so we've now moved from these sort of simple model engineered tumors to several different things. We and our colleagues have worked on deploying this in glioblastoma, ovarian cancer, and pancreatic cancer. And there's several different things that we have learned about the SynNotch CAR-induced circuits and how well they perform versus solid cancers. One is that the cells kill locally. They only kill where the priming and killing antigen are in proximity. And importantly, and I won't talk about this in great detail, but CAR expression shuts off once the cells leave the region of priming with a half-life of a few hours. So they don't have enough time to really mount this big proliferative multi-day response that you need to clear a tumor. In addition, it's important to note that the priming antigen is now only used for recognition and not for killing. So it's really just an informational molecule that localizes where the killing is going to take place. And importantly, it doesn't need to be homogeneous. It doesn't even need to be on the tumor cell. It can be on neighboring cells that are part of the tumor. And what that does is it creates this blast radius where the cells that are primed now can kill any cell that has the killing antigen over a blast radius. And that's important also because then we can harness things like neoantigens that are very specific but heterogeneous as a priming signal. It still can be really important in the recognition. The killing antigen need not be perfectly specific as long as the dual antigen combination is only found on the tumor. That's important because it means that we can actually start if we prime in a very localized way, we can start killing based on a broader set of antigens, including ones that that might have cross-reaction in other tissues, as long as those don't occur with the priming antigen. And then, importantly, conditional induction of the CAR versus constitutive expression greatly decreases the degree of CAR T-cell exhaustion and improves the durability and the tumor clearance. Many of these circuits are much better at clearing these tumors in the circuit context rather than as a constitutive CAR. And then also importantly, and I won't talk about this, but it's a very important part of this, is that SynNotch can also be used to locally induce inflammatory payloads. We've done this with IL-2 and found it to have a really powerful effect on overcoming the suppressive tumor microenvironment, both in syngeneic pancreatic and melanoma tumors. And it is more effective than armored forms of CARs that are constitutive or linked to the CAR-induced expression, which is also could be accelerate off-target effects. So I want to talk about, we've done a lot of this work in collaboration with my colleague in neurosurgery at UCSF, Hideo Okada, who's an expert in glioblastoma. And there are two SynNotch CAR circuits that we've been developing in glioblastoma. One is we call eSync, and that is a SynNotch circuit that primes with a tumor neoantigen EGFRV3. That is a splice variant that's on that changes as part of the extracellular domain. You can recognize it with an antibody. This is very tumor specific. But when they did a trial of using a CAR that recognized it, it went to the tumor, it killed the EGFRV3 positive cells, but because this antigen is very heterogeneous, the tumor escaped and survived with editing of that antigen. So it's very powerful, but we want to use it not for killing but in a selective way, but as an information sort of priming antigen. So we've done that, and we've used it to prime, as a SynNotch receptor, to prime expression actually of a two-headed CAR for FA2 and IL-13RA2. These are two antigens that are actually more homogeneous, but actually imperfectly specific. But by combining them in this particular way, where one gives more complete killing and one gives more localized specificity, it works really quite well. This has been published. This is now, we have an IND approval and are now recruiting for a phase 1 trial, so we're very excited about that. I'm going to talk a little bit more about this other circuit, which we call BSync, which is priming on a normal brain-specific antigen. So BCAN, as I'll explain, is something that's only found in the brain. We can make a SynNotch that recognizes that molecule in the brain and then induces this two-headed CAR, the same one. These antigens actually are not expressed in the normal brain. So it ends up being really very specific, and we're very excited about it, and I'll tell you about that. So one of the key concepts here is that we'd like to actually be able to restrict a therapeutic, not to just recognize its molecular target, but also restrict anatomically in a tissue where it acts. It's hard to do that with a molecular drug, and molecular drugs, as I said, will often bind with multiple tissues because the same or related targets are in different tissues. And let's say we wanted to have this attack something in the brain, then one, it might have trouble getting into the brain, but also would cross-react with these other sites. Now, if we could, say, magically instruct a drug to only go to the brain, it would solve a lot of problems. It might be more effective in the brain where this disease is, but then also we would drop out all the cross-reaction that occurred outside of that tissue. Okay? And, of course, this is how a lot of biology works. Things go and occur in specific tissues, and that's why the same molecules can be used in different contexts. So how might we actually try to design a cell that could restrict its activity to a specific tissue like the brain? So what we've done is we've tried to identify brain or CNS-specific extracellular antigens that we could recognize, and then design SynNotch receptors that could recognize these brain antigens, and then used an anti-brain SynNotch T cell to induce expression of a therapeutic payload. Now, in the case of a cancer like glioblastoma, we could induce the expression of a CAR, which would only kill within those antigens within the brain context. But as I'll tell you later, we're also interested in expressing other kinds of payloads for other brain diseases. For example, in neuroinflammation, could we express suppressive payloads? Okay, so we did, with Olga Troinskaya's lab and Yan Wang, who's a student in her lab, we did an extensive search to try to find the most specific molecules, and there are certain molecules in the brain that are extracellular ones that are quite specific. That includes certain surface molecules that are on neurons or on oligodendrocytes on the myelin sheath. But one of the ones that came up were a number of molecules that are part of the unique brain extracellular matrix. So there's a these perineural nets and other things in the brain. There's a very extensive extracellular network. It's specific. It has hyaluronic acid and tenacin, but then there are these chondroitin sulfate proteoglycans that are very specific to the brain, especially this one BCAN. So we worked with DevCedu. We raised antibodies against this and made validated BCAN SYNNACH receptors. And this just shows BCAN, Brevican, is made by many cells, including astrocytes, and secreted into the ECM. This is a T cell that has a SYNNACH driving a GFP, and as you can see, as it interacts with the region around the astrocyte in red, it gradually turns green. So we can actually get these cells to turn on a response to BCAN, and we've shown that in a number of cases. So how do we apply this to something like glioblastoma? So I just want to review again. The antigens that we've chosen to use for killing are well-known glioma-associated antigens like FA2 and IL-13RA2. Both of these are well expressed on most glioblastomas, but one of the problems is that these are also expressed in other normal tissues, but not in the brain, not in the normal brain. So what we decided to do, and this is work from Milos Simic, who's leading this work, is to make a SYNNACH against BCAN and have that, so that would say if we detect the brain, now we'll turn on the CAR that will kill these glioma-associated antigens, even though they're not absolutely specific. But they are the only ones in the brain, and so the idea is that the intersection of these two Venn diagrams would be just the glioblastoma. Milos and Payal in Hideo's lab went ahead and they made these cells, tested them in vitro, and then tested them now in vivo with a GBM tumor PDX model that was implanted in the brain. And what we found is that this really very robustly cleared these tumors from these and increased survival. Nearly all of the mice survived. This is one of the strongest responses we've seen of all of our glioblastoma therapies. So it was acting very effectively, and we were getting a lot of priming. And this is shown here. So we now go take the tissues early, mid-treatment, and we look at the CD3 T cells. We see that really there's only significant activation of the SYNNACH in the brain sample. Okay. We're very excited about that. It seems to be very strong and brain-specific induction of the SYNNACH. And one of the things that we're most excited about is the idea that this could really be a pan-GBM therapy. It's not that we have to find patients that have that specific neoantigen to prime. This should work for all patients. And that's a big deal in terms of thinking about how to screen and how hard that is for finding the right patients. Now, what about outside of the brain? So what we did is we took the same tumor cells and implanted one in the brain and then one in the flank. So they both have the killing antigens, but only the one in the brain will be surrounded in this environment that has the brain priming antigens. And what we see in this case, in the same mice, is that the brain tumors are cleared, but then the flank implanted tumors are not cleared in those same animals. So it really indicates that this is brain-restricted activity. Okay, so we now actually are really excited that we have this kind of GPS system where cells can know that they're in the brain, and we actually have some interesting results about some things in lung and pancreas also. But what we realize is that maybe this could be used for other diseases where we needed to locally deliver things to the brain. And neuroinflammation was an obvious thing. Now, there's been a lot of work in the past of people trying to find different cytokines or factors that could have impact on neuroinflammation, particularly in EAE models. And some work was shown really, I think, over 10 years ago that something like IL-10, a suppressive cytokine, when expressed in the brain, when you inject AAV in the brain, that high-level expression can block EAE. But, of course, systemically injected IL-10 is ineffective. It's either not able to get to the brain efficiently or not enough at a high enough level. So Milos, working with colleagues of ours, including Scott Zamville in the neurology department, then tried to ask, could we make a T cell with a brain SYNNACH deliver IL-10, and how would this fare against EAE models of multiple sclerosis? And so let me just briefly tell you about the model. We used animals that were primed against MOG, which is an autoantigen from the myelin sheath cells. And you can obtain CD4 T cells. We polarize them to Th17 and then adoptively transfer that into a mouse, and that mouse will develop neurodegeneration. and neuroinflammation. And so in this case, we would now ask, could we transfer the SynNotch IL-10 therapeutic cells, putatively therapeutic cells, into those cells, and would those have any effects on the EAE? So we look at these different effects, neurological scores of paralysis, et cetera, and what we see is that, in fact, when we give these cells, we can see a significant amelioration of the severity of EAE and much longer survival of those cells. And this just shows some of the mice where the top are ones that were untreated. They developed EAE by day 12, but at the same time, those mice that were treated with the suppressive cells really maintain fairly good mobility. So we were able to see an effect there. And so we're excited. This is very early days, but this idea that we can target cells to tissues. So just to summarize this part of the talk, we've engineered T cells that have used that use multi-receptor circuits, multiple components linked together, now to integrate information at different scales and to make precise disease-specific decisions. As an example, in the case of glioblastoma, we're able to engineer precise brain cancer cell therapies by programming cells that can, one, sense that they're in the brain, and then two, induce expression of a CAR. And this is an example of a powerful combination of anatomical and molecular specificities that really only a cell can do effectively. We are excited that we have developed a way to have brain sensing cells that can be used in a disease-agnostic manner to deliver either immune suppressive payloads for inflammation as well as ones like CARs for killing cancer, but potentially also for regenerative payloads or ones for neurodegeneration. I should say we're also working on trying to improve, say, cell migration transfer migration across the BBB and other trafficking things that could help further, but also it's possible that we could target other tissues in an analogous kind of tissue GPS, cell GPS mechanism. And I just want to remind everyone that we're really excited that we are starting this EGFRV3 SynNotch CAR trial now with this team at UCSF, as well as in midstream in terms of preparing our IND for the brain-primed BCAN1. So hopefully we'll be able to report on that soon. Okay, so the last part of my talk is I want to talk about trying to apply these same fundamental synthetic biology tools to a different problem space in biology, but one that still requires cells to talk to each other in very precise programs. And that's in the case of development, how cells interact with one another to change their fates and organize in space, and how that kind of complex behavior can be kind of pre-programmed into a set of DNA. And so this, what I'll show you in this movie is actually some very simple cases where we've taken a fibroblast cell line and engineered novel synthetic programs and got them to organize into different kinds of things in a spontaneous way. Okay, so why engineer development? Again, we're interested in this kind of from a bottom-up view. Are there kind of core operations that are utilized in different developmental programs, and can we understand how they're put together to construct all the different kinds of morphologies that we see in metazoans and different organs? And can we also learn to program new or altered morphologies? Might there, for a transplant or something, might it be better to have a heart that has certain properties that are unique to that? And so the fundamental question here, as part of what we're trying to do at a more basic level, is to ask, can we use the same sort of toolkit that we've been using for engineering the T cells to also engineer development? Are those really the fundamental relationships that are powerful for creating these new programs? And so several years ago, Satoshi Toda and Leonardo Morsuit, when they were postdocs, they both have faculty positions now, they started to try to ask, could we take cell lines like L929 fibroblasts that don't self-organize, and could we put in programs that would? And that asks, then we have to understand, what's the basic logic of developmental programs? And one hypothesis was that at its very core, most pathways in development have cells that talk to one another, and then those induce selective changes in morphology. So the idea here was that could we use SynNotch to engineer new relationships between cells to recognize one another, but then link those to the induction of various morphological outputs, things like adhesion. Could we induce the expression of cadherin molecules or other adhesion molecules that would change how the cells sort? Could we, as a stand-in for differentiation, could we induce fluorescent proteins? But could we also induce new signals, like new SynNotch ligands that would now carry on this conversation even further? Okay, And so they did that, and this just shows some of the things they were able to do. This is a cell population that is able to interact with one another, and then you get this differentiation into a green and a red population. In this case, they organized in this polarized fashion. And then the next example is one where we have a similar set of cells that we put together, and then they start interacting, and what you'll see is the formation of a green-colored core, and then that leads to the formation of an intermediate red or M-cherry intermediate shell. And just to kind of explain how this works, in this last case, this really is this reciprocal signaling or conversation between two cells. We have two cells, A and B, shown here at the top, and what the A cell has a signal that will turn on a SYNAPse in cell B that will turn on both GFP as well as a high level of cadherin. So you get green cells that now sort to the middle. It turns out that GFP we used was an extracellular version. So that actually, we have a SYNAPse in cell A that recognizes that and turns on M-cherry and an intermediate level of E-cadherin. So those cells turn red, and then they form this intermediate shell. So this happens in a very spontaneous and stereotyped way, consistently. We can cut them, and they'll still regenerate that structure, et cetera. So we can get kind of simple self-organization. This is another example of, a lot of development has larger scale morphogen gradients that lead to patterning. Satoshi showed that he could make a SYNAPse version of a morphogen, a fusible signal, by making secretor cells that would produce a GFP in this case, and then have in what we'll call a body, both anchor cells that would trap the GFP and then could allow it to be detected by a cell that has an anti-GFP SYNAPse that could turn on, say, M-cherry. And this just shows a setup where we have a set of GFP senders and then a body of cells, and this is 4 millimeter scale, so fairly large scale. What we see is that we're able to generate a gradient of response that emanates from that source. And if we now elaborate that with various positive and negative feedback loops that occur in the body, this artificial body of cells, we're able to get that same body to respond in different ways to those gradients, and we get different kinds of patterns like shown on the right side. So we can get kind of a French flag pattern, the classic kind of responses that have been theorized in developmental biology. Okay, so I won't talk, much of that's been published, and there's a lot of interesting stuff there, but that's kind of taking cells that don't develop and trying to make simple things from scratch. What about trying to actually guide the development of cells that have the potential to develop in different ways, like stem cells? So one of the things we've been interested in more recently is asking whether we can take ES cells, in this case we're working with mouse ES cells, and then can we engineer cells that will talk to them in spatially defined ways. And these are what we're calling synthetic organizer cells that can provide morphogenetic instruction. So, and what I'll show you is that we can get some pretty interesting things to come out of that. Okay, so the idea is that when we look at development and we try to drive differentiation in vitro, most of the time we're taking progenitor cells, ES cells, and we are adding morphogens to the media. Maybe we're changing the time and the dose, et cetera, but we're doing it in this isotropic way. And that's very different from what happens in vivo, where there are cells that are in specific locations that are producing various morphogens in a spatial temporal specific way. And some of the most exciting work very recently on these synthetic embryos is this work from various labs that show that extra embryonic cells can often self-organize around ES cells and provide the kind of spatially specific signals that lead to quite advanced in vitro embryogenesis. So can we actually try to recapitulate that? Could we engineer synthetic organizer cells that would organize in space around the ES cells and then also be now the sources of these morphogens? Okay. So we need to be able to program spatial self-assembly, and this is where we turn to both natural adhesion molecules as well as some of the engineered ones that we can program with these synthetic adhesion molecules. And as I showed you before, these movies I showed earlier are actually fibroblast cell lines that were engineered, the same ones, but engineered in different ways with different adhesion molecules to organize around these red ES cells in either forming a node or a shell. And what we can do with these is now we can combine them in different ways, and we get different kinds of architectures, single nodes, dual nodes, shells, nodes and shells combined, so we can get different kinds of patterns and we can now try to express morphogens from these in anisotropic ways. So the first thing that this is work from Toshi, a postdoc in the lab, is to ask, could we deliver this key early embryonic morphogen Wnt3a from specific organizer cells? And what we find is that we can generate very tunable and directional gradients. So these are ES cells that have a Wnt activity reporter, or PTCF M cherry, so that's what it's going to turn on. If we give a Wnt agonist chair in the media, you see it turns the whole thing on. If we give Wnt in the media, it turns everything on, at least in this isotropic spherical way. If we provide Wnt, we make it, express it from this shell, we see something very similar. But if we now express Wnt from this node, what we see is this polarized activation or gradient that goes across that, and then elongation of the body along that, what looks like a primitive anterior-posterior axis. And also, if we now actually make a second node and put it 180 degrees away, and now have that make a Wnt inhibitor, DKK1, we can get an even sharper gradient, a much steeper gradient that covers the range from very high maximal activity to low activity across the span of that body, if you will. So what happens as we tune these gradients? What happens to the morphological outputs? So what's shown here is just after 8 days, some of the things that we see. And so I want to start with the middle one, which is where we have both the Wnt and the DKK nodes, the inhibitor. And this leads to this very steep gradient with a high dynamic range across the whole body. And what's interesting here is that what we see is that we see specific tissues arranged in more or less the correct anterior-posterior arrangement. We see things like OTX2 neural markers occurring near the DKK1 node, and then various markers. But for example, this is cardiac tissue, very localized here in the middle. And so we think this is the anterior and the posterior in this dual node structure. Interestingly, though, if we just use a single node with DKK1, we get mostly almost all neural fates. If we, on the other side, go and have this Wnt node alone, what we see is a shallower gradient. We don't see the anterior type structures. We just see posterior type fates, and the cardiac tissue is at the most anterior there. So what we see is that we can see specific lineages arising, and they're organized in the appropriate anterior to posterior topology. But the gradient properties, the range and the shape and the steepness, determine the spectrum of lineages that we observe in these resulting embryoids. This just shows one of those ones with the dual heads that has the most range of fates. One other thing I just wanted to point out, I hope you can see in this movie here, is that this is the beating cardiac tissue here. It's really localized. It's not really dispersed. These are self-organizing into very localized subtissues in this anterior-posterior axis. Now, one of the more interesting things, if we look more carefully at the case when we have just a single Wnt node, as I said, what that happens there is this gradient actually is less steep, and you don't get all the tissues, but it's shallower. And what we find is that we actually get very interesting morphologies suggesting that this has kind of higher resolution spatial information or morphogenic information. One of the things that we see in a significant, probably about 15% of those embryoids, is this beating heart-like chamber. We see a cavity inside, and this heart that, by calcium imaging has a very regular beat, which was quite unexpected and really difficult to see in a spontaneous, self-organizing way, this kind of structure. Most other efforts at kind of reconstructing hearts have involved taking multiple lineages and combining them in assembloids. And the other thing is here, not only do we see this heart-like cavity, and it has markers like cardiac troponin or the transcription factor NKX2.5. Those are localized to the cardiac region around the cavity. But also we see by staining for endothelial markers that we can see this looks like it's linked to this extended vascular network marked in green here. So the shallower gradient yields higher resolution tissue structure as a result, and really this is a really amazing case of self-organization of a multicell type heart-like structure in a spontaneous way. So one of the things that we're thinking is that there are kind of different ways of looking at how to drive development in vitro or reconstituting it. Some are most work done in the past has been about kind of taking ES cells and using combinations of protocols of differentiation factors to drive differentiation of specific lineages. And maybe you combine those if you're interested in tissues. On the synthetic embryo side, using fairly natural cells like the extra embryonic cells that talk to them. And the idea is really, can you push all of the lineages and get the self-organization of the whole body? We think that with some of these synthetic organizers, we may be able to kind of tune somewhere in between that to drive certain regions or ranges of lineages and still get some self-organization there, or even to drive things to non-natural, non-native fates in some controlled way. So again, this is very early days, but we're very excited about that. So what we've shown here with this synthetic development work is that we can combine components that control cell-cell signaling, differential cell adhesion, as well as morphogen production to engineer self-organizing multicellular assemblies. These are programmable and autonomous. They can generate distinct but highly stereotyped morphogenic outcomes. And that in particular, synthetic organizer cells can provide progenitor cells with information-rich spatial gradients that could generate complex tissues, like a localized heart-like assembly that beats, has a central cavity and an associated vascular network. We can custom tune the gradient range and steepness to alter the range and resolution of these morphological outcomes, and that this approach, we think, could be applied to many different types of morphogens as well as progenitor cells. And one of the things that I think is really tantalizing sort of future possibility is, could we use this kind of approach in vivo to generate local morphogenic gradients, or to reconstruct them around sites of injury or other to drive regeneration in situ. Again, a long ways away, but really exciting. So really, to conclude the whole talk, I hope that I've been able to show you that I think living cells are these amazing machines that, unlike sort of other scales like molecules, have this much higher level to integrate information over a hierarchy of scales, be that anatomical or molecular. And they can spatially coordinate their activities by sensing and interacting with their environment or their other neighboring cells. And that if we take advantage of this, there are many things that we can do, and we can build tools and sensors that allow us to very precisely control and change these cellular interactions, and we can use them to build different kinds of cellular programs and circuits. These include using these to operate in many different cells, including immune cells that can search for cancer, or can target tissues for suppressing inflammation, or putting them in cells that can provide spatial instructions that can guide development. And just want to leave you with the thought that understanding how to manipulate the logic of these cellular systems really will answer, I think, a lot of the questions in biology that we don't understand. We know a lot about the components of biology. We know less about how they are put together, the logic of that. And I think this provides a way to both explore those questions, but also powerful ways to generate new therapeutic platforms that can interface with complex diseases like cancer or fibrosis or autoimmunity, where we really have to have a cellular agent that can participate and redirect the conversations that happen in these ecosystems. And so with that, let me thank all the people in my lab and my collaborators. I want to specifically focus on a few people working on the brain-targeted cells. Milos Simic has been the fellow who's leading that, together with Payal Watchmaker, but there's a collaboration with Hideo Okada and Scott Zamville, both at UCSF in neurosurgery and neurology, but then also the bioinformatics was done in collaboration with Olga Troyanskaya, my colleague at Princeton. And then the synthetic development was really spearheaded by Satoshi Toda and Leonardo Morsut, but the synthetic organizer work was done by Toshi Yamada and Coralie Transisau in collaboration with my colleague Ophir Klein. So thank you very much.
Anyone with questions, please use the mics. We do have a question that came in, if you're ready for it.
Okay.
So how do brain-targeted CAR cells penetrate the blood-brain barrier?
Yeah, okay. So the great question, the question is how do these CAR T cells penetrate the blood-brain barrier? Well, so we know that there is always, T cells can get in the brain. They end up causing MS. They can get in some of those early GBM trials. That EGFR-V3 CAR, they did get into the brain. I think that it's pretty clear that in diseases like brain tumors and MS, there's also some damage to the BBB that allows cells to get in earlier, more effectively. So, it's unclear. Our cells are getting in there. We do see them. They're proliferating. So they do get in to some degree. We think it's amplified in these particular disease cases, but we are working on trying to improve ways to get them through the BBB.
Okay.
Okay.
I wonder, also very, very elegant and brilliant as usual. So my question is about the specificity of your GBM targeting T cells. You showed some background activation in the spleen.
Yeah.
Do you understand it? Is there enough ligands in the spleen? What's happening there? Is it some leakiness in your syn notch?
So you're seeing the glass half empty.
I'm French,
or half
right?
Full, whatever. Yeah.
No, I'm very concerned about background and specificity.
Yeah,
right?
I mean, it's much lower. There's a little bit. I would say, I guess, we've done extensive non-brain tumor killing, and we don't see anything there. We don't see proliferation of the cells there. So, we think we're below the threshold for any sort of effects. Yeah.
So I guess that's my more general question. Moving from the mouse model to the real patient, do you have a way to test the different tissues to see what?
Well, I mean, we've gone through the IND process on this, and we are using mouse cross-reactive antibodies. We're doing tox in the mouse. We're also checking everything against human tissue libraries, and so far that's all checked out. So, and this is what a dose escalation is for. Yeah, I would say though that a lot of these, the EGFRB3, that CAR has been in trials, and there wasn't a problem. So that a combination of things, but there are unknowns. Yes.
When you were showing us the expression of the Wnt, and you showed us the gradient when you induced the nodes and the inhibitors, what were you using to direct the nodes and inhibitors on those?
Oh, you mean to the second node?
Yeah.
Yeah, the second node is a little bit, has a more stochastic element to it, and we're trying to improve that.
Okay. And on the cardiac expressing cells, what were they? So they induced the vessel expression in the nearby cells. What did they do? What do those vessel expressing cells do to the neighboring cells?
Well, okay, we are trying. Yeah. So we're just kickstarting the event, right?
Okay.
This is one signal, and clearly, there's a lot of Wnt, BMP4, a lot of things that go on in regions there. So we are trying to track, by RNA-scope and other things, what's the sequela of events that happens? Because yes, of course, development is, and this is the tricky thing about it. It's one more vision kind of triggering the next thing, and so forth. So that's all to be worked out. Yeah.
Thank you.
Yeah, great talk. I have a question about your brain-specific synaptic CAR, and you are using tandem CAR, and I'm wondering if there's any difference when you have only single target versus two targets, and any rationale behind using tandem CAR? Yeah,
I mean, the rationale behind using the tandem CAR has to do with the fact that every patient is different, but the number of patients that have high, relatively homogeneous expression of this combination of two is higher. So when we do like an AND gate where you need two things, it's really the intersection of a Venn diagram. If you're looking at a tandem CAR, that's an OR gate. That means you're now killing across both circles, right? So we want to make the decision switch of specificity to be very specific, and then we want the killing to be broader.
All right, thanks.
Yes.
I wanted to build off of the question on the synthetic organizer cells in the assembly, which is incredible that you can see the development of the cardiac tissue and things like that. Yeah. But could you combine that with your SID-Notch system to further direct development in a specific way once you know what the cell type is, to have cells that have.
Yeah, no, that was really our intent at first, but then we realized, well, we got to do first things first, and it was even just localizing them would be very important. But yes, we have now the ability to kill nodes and replace them with new nodes. And what would be really powerful when you try to string together these different events, as in development, is to be able to now have an organizer that would detect a marker of the next stage of development in a localized way. So we're totally into that.
Thank you.
Hi, Professor Lim. Really fantastic work. I kind of have a naive question. So if we, other labs want to use SynNotch system for some customized in vivo experiment, so how could we utilize that? Are those SynNotch systems a bunch of plasmid with multi-cloning site, or more ideally, are there some companies we can work together for pay for service?
No, that's a great question, and I would say that there are now several versions of SynNotch, and also we found that when we take a new antibody, an antigen, that there's some degree of optimization that we need to do, and it also depends on some versions produce more payload but have more leakage. Some are tighter but have less, and there's also cell type specific things. So one of the things that we're trying to do is come up with to make our lives easier, but also everyone's life easier, is to come up with a small library of different templates where one can pop in your antibody, your SCFE of interest, and then test those, and hopefully guide people to think about what the application is and what behaviors would be the best. Yeah.
So is this like a mini library available on AdGene?
Not yet. I mean, a lot of these things are available on AdGene, but now we're doing things like changing the height of the extracellular domain and different scaffolds for the receptor. Just saying we're expanding that for our own case, but we'll make that available. Yeah.
Okay, thank you.
One more.
Thank you for the talk. I have a very general question. I'm thinking whether there are any emerging phenomena in the work you presented, unexpected cell states or unexpected cell functions.
And are you talking about in the T cells or the developmental systems?
In both.
Yeah. Well, that raises obviously a great question of what are there possible cell states that you can drive cells into, be they immune cells or stem cells along some trajectory? And I suspect that there are. I think that's a very fundamental question. You see a certain realm of stimulations during life and evolution, and are the cell fates of the T cells and the stem cells so canalized that they stay on track or not? I think it's pretty clear from RNA-seq and whatnot that they can go off some. There are certainly cells that are hard to classify. Okay, to what degree I don't know, but that's a very sort of important and fundamental question.
Thank you.
Okay, thank you very much, everyone, for coming to the lecture. And this has been a really, lively questions and answers. But you can continue, chatting with the speaker if you want. We have a reception just outside the auditorium. And with this, this wraps up the lecture. Thank you, everyone.
Thank you.